Okay, so welcome back to the second part of our morning session in the Bio-Ontology COSI. While the first presenter is setting up, let me just summarize what we're going to do. So please, we are going to have a number of presentations, starting with a Proceedings presentation. And we are going to have our closing session at 12 or 10 minutes past 12 o'clock. And all of you who are interested in how this COSI is going to evolve in the future, it would be good if you would join us at least for the closing session, because there are some changes which are happening now with how the COSIs fit into this conference and also the society. So it's something that we as a community should discuss. Okay, so we'll start with our scientific program. Please allow me to welcome Wenhui Xing. Wenhui Xing, and she will give a Proceedings presentation titled A Gene-Phenotype Relationship Extraction Pipeline from the Biomedical Literature Using a Representation Learning Approach.
Okay. Ladies and gentlemen, it's a great pleasure to present my research here and to get your feedback. My name is Wenhui Xing. I'm currently a PhD student at Wuhan University of Technology. In today's talk, I will introduce my ongoing doctoral research on a gene-phenotype relation extraction pipeline from the biomedical literature using the representation learning approach. One of the most important topics in modern biology is to establish gene and phenotype relationships. With repeatedly growth of the biomedical literature, a lot of unstructured information is implicit in them. Recently, natural language processing and text mining technologies have been widely used, also in relation extraction, like the protein-protein interaction, drug-drug interaction, gene-drug mutation relations. However, the important issues of gene and relationship extraction is really inadequate. Currently, there is lack of good open source algorithm and research, especially for plants. And many researchers are still following the traditional expert identification method, and that is very time consuming. The speed of expert verification is far less than the explosion of the literature. And how to minimize the expert verification and to achieve the relation extraction is our concerned problem. For these reasons, we want to design the gene and relationship extraction pipeline. This pipeline contains two main parts: the name entity recognition and the relation extraction. In the name entity recognition, we need to identify gene and a phenotype. Because genes have contextual regular patterns in the literature, so we can use the rule-based and the lexicon-based method. For the relation extraction, it also exists some algorithm like the text runner, reverb, OLIA from the OpenAI system. So the challenge is the phenotype recognition. More concretely, the phenotype recognition challenge of it is 3 points. First, because phenotypes are descriptive information in the literature, so they have a complex boundary problem. It's sometimes not just a word. It may be a phrase of different lengths or even a sentence. Next, similar phenotypes may describe different by different authors and literature. Like this example, a leaf curly and curly leaves. They are the different description in the literature. Moreover, there is lack of phenotypic lexicons, especially for plant phenotype. So there is no annotation corpora to help learning or training, making it difficult to use a supervised or semi-supervised learning approach. So the phenotype recognition is a bottleneck of the relation extraction. Therefore, we want to design unsupervised representation learning approach for the phenotype recognition. This is the architecture of the relation extraction pipeline. First, we downloaded large amount of literature from PubMed, and then we designed and supervised the word embedding Cascade sentence embedding algorithm to perform the phenotype extraction. After the Cascade approach, we found that some sentences, descriptions of phenotype, have regular contextual pattern. So we designed the phenotypic sentence template algorithm to discover this pattern in order to expand the recognition. Next, we use the external database for the gene recognition to identify gene IDs and the gene names. After the name entity recognition, we modified the OLI algorithm to perform the relation extraction. Next, I will give you the detail of every part. The Cascade approach of phenotype recognition are shown in this slide. First, we trained the word embedding model using preprocessed PubMed literature. We using the external database, using the external ontology database from the ontology, plant trait ontology, and AHD phenotype ontology as terms as seed to find high similar phrases in high-dimensional vector space. In the same time, we also recognized and revised the abbreviation to reduce the error rate. Meanwhile, regarding the phenotype-containing sentences as the positive samples, this word embedding method can find frequent phenotypic phrases, as many as possible. Due to the complex boundary problem, in order to identify long or short phenotypic sentences, we cascade the word embedding results to the sentence embedding. And we designed the negative class label enhanced algorithm to modify the sentence embedding model as the weekly supervision model. The purpose is to expand the diversity of recognition. Finally, using the positive samples as seed to find high similar sentences in high-dimensional vector space as the phenotypic sentences. After the Cascade approach, we found that large-scale gathered sentences have similar sentence contextual structure. Like this example, these two sentences describe the phenotypes have the similar structure, like B required preposition. So we designed the phenotypic sentence template extraction architecture algorithm. It used the statistical combination and the mechanism of solving combination explosion. With this algorithm, a sentence template is considered as an element ordered sentence word. And this algorithm generates all possible combination and selects the frequent combination as the pattern. And this pattern must contain the phenotypes. We also show you some examples at the bottom of this slide, like the play something role in, regular regulation of, and involved in. The gene recognition is not very difficult. We can use the external database like the UniProt, NCBI for the lexicon-based method. However, some gene names are not in these lexicons, so we can combine the character level rules and the contextual level rules to improve the gene name recognition. For the relation extraction, we use we input the collected gene and phenotype co-occurring sentences into the OLIA algorithm. Because OLIA gives non-phrase pairs of sentences as paragraph, and these non-phrase pairs contain too many noises. So we modify this step. We mapped the first and the third argument to our previous phenotype and gene entity list. And if one or some gene and phenotypes are in each of these two arguments, we consider these relationships is the gene and phenotype relationship. To evaluate our pipeline, we designed two types of compression experiment using model organism Arabidopsis thaliana as experimental object. The first is according to different phenotype recognition and relation extraction methods we compared with baselines. The second is our pipeline applied in the entire data set. We compared coverage rate. This table shows the performance results of our pipeline algorithm and the baseline algorithm. And from the table, it can be seen that our pipeline algorithm can not only have a higher precision, but it can also effectively improve the record value. That's because our Cascade representation learning approach can expand more than 2,000 phenotypic expression and increase 2.9-fold compared to original ontology, thereby increasing the final F1 value. When comparing with TIRE, we first process the original data set, and we can get the 481 full papers and 1,397 sets of gene and phenotype relationships. We apply our pipeline to all these papers, and the results have covered 70.94% original data. And in the same time, we can identify 373 new relationships. Using our pipeline, we can automatically update the gene and the phenotype relationships as the literature growing. Although our pipeline can effectively improve the final relation extraction, there are also some limitations. First is the error of relationship extraction. Because the OLIA just identifies relationships in single sentence, and the longer sentences distinctly reduce OLIA accuracy. However, some biomedical literature sentences have more than 13 words. It may cause some inaccurate results. And in the entire data set, some relations are cross-sentence or even cross-paragraph. It's very complicated, and we cannot process this type of data now. So this is a main error of our pipeline results. Also, the inaccurate phenotypic boundary and the problem of gene recognition also cause some error. In the entire data set, some phenotypes are not the descriptions in original literature after manually processing, so we cannot cover this type of data. Now I'll give you the conclusion of our work. We propose a pipeline of gene and phenotype relation extraction. It can significantly expand the original phenotype ontology using our cascaded representation learning approach. Our pipeline obtained an 66.83% F1 score that outperformed other baselines. And applying the pipeline to the entire dataset, we can complement 373 new relationships. In the future, we may consider the event extraction, like including the environmental influences, instead of just the binary relationships. Now I want to thank my advisor, Professor Dr. Xiaohui Yuan, and my vice advisor, Professor Dr. Lin Li. And also I want to thank all of our used resources like the PubMed, PTO, AHD, Uniprot, NCBI, TIRE, and many other tools we used. They are very brilliant, and they help us to perform our research. Thank you for attention. Thank you for listening. Thank you very much.
So thank you. We have time for a few questions. So I will make a start then.
Okay.
So you're using the plant trait ontology, and the plant trait ontology contains, or is an ontology of traits, not of phenotypes. So you will find things like leaf size, but you will not find smaller leaves.
Okay.
But you showed that you find smaller leaves, or that you find reduced growth.
Yeah.
But in PTO, you will not find this. So how do you get to the phenotype when you start with PTO as a trait?
The PTO just a part of our work. We also use the Cascade, the word embedding and the sentence embedding this approach. And this approach uses the original term, ontology term from the PTO AHD, and Z can use using our word embedding algorithm. Z found the new expression, not the original term from the ontology. So the ontology help us to perform our algorithm.
And would it not work better if you take a phenotype ontology as a seed? Like if you take plant phenotype ontology or FLOPO, an ontology of plant phenotypes as a seed, would this improve your performance?
Yeah. Oh. Okay.
Okay, well, thank you.
Okay, thank you.
Yes. Have you tested how the performance of this would act if you used multiple ontologies? So from my background, I'm looking at the human phenotype combined with the disease ontology. Because in human disease, many papers often just mention the disease and not actual phenotype terms. And also in the human world, it seems that they blur the lines as to what a clinician would call a disease versus a phenotype. They can go either way. So do you know how this might perform in that situation?
We just focus on the plant domain. So maybe we didn't use the HPO, the human phenotype ontology like this, but we will consider to use this later in the future work.
Okay.
Okay, thank you.
Okay.
Thank you so much.
Another part of this might be to extend it to include the cultivars or germplasm that's involved with these interactions, because if you're looking at Arabidopsis, there's a lot of ecotypes. You can, so really the important part is the plant microbe interactions that I think a lot of people will be looking for eventually. So, that might be a way to extend this work even further. So.
Yeah, thank you. So you think we need to also process our original data to the structured?
Well, yeah, for instance, I work with crops, so a big interest is disease resistance, and a lot of research papers look at host differential reactions.
Okay.
Is it susceptible? Is it resistant? And then they're also looking for the genes involved in that. So that may be one aspect to concentrate on or extend it.
Yes, thank you. Thank you for advice.
You did mention that you discovered more than 300 new relationships using your approach, which is really impressive.
Mm-hmm.
Can you tell us more about the relationships extracted, and did a human expert look at them and kind of do like, okay, is this really can it be validated of sorts? Like, okay, these are valid or not?
Actually, in some literature, they did not write so clearly about the relationship between the gene and the phenotype. So nowadays our work just more about extract this binary relationship, and the next step we want to figure out the exactly relation, like the increase or some inhibit or some regular something like this. So I ask you a question. I answer a question.
Yeah. Thank you.
Thank you.
Mm-hmm. Any other question? I have one last question. So, is the source code for your pipeline available, and are the embeddings and the predictions that you make, do you have them on GitHub somewhere?
Yeah, this source code is available. You can search the paper is a publication, and we have the HTML URL HTTP, you can download the pipeline source code.
Okay, so thank you.
Thank you.
Okay.